Hi, and welcome to my presentation on the diversity of microbial eukaryotes. So before we get into the major groupings of eukaryotes, we need to talk again about endosymbiosis a bit. So we had already gone over primary endosymbiosis in earlier chapters. Primary endosymbiosis is how eukaryotic cells gained mitochondria and chloroplasts from bacteria. So you had a bacterial cell that was free-living, that was engulfed by an early eukaryotic cell, and instead of dying, it turned into an organelle. You had a proteobacteria that was engulfed by a eukaryote and turned into a mitochondria or a mitochondrion, and you had a cyanobacteria that was engulfed by a eukaryote and turned into a chloroplast. So today all eukaryotes have mitochondria. They're all descended from the early eukaryote that got those mitochondria by endosymbiosis, and also every photosynthetic eukaryote is, or I should say not every photosynthetic eukaryote actually, but all of your plants and your algae are descended from the early eukaryote that got chloroplast by endosymbiosis of a cyanobacteria. So that's our two major endosymbiotic events that kind of were essential for establishing the eukaryotic lineage. But we've also had additional endosymbiotic events that occur throughout kind of the early evolution of eukaryotes, which would count as secondary endosymbiosis. In secondary endosymbiosis, instead of a bacterial cell being engulfed by the eukaryote, you have an actual eukaryote, some type of algal cell, being engulfed by a bigger eukaryotic cell. So these algae are kind of swallowed up by these larger eukaryotic cells, and instead of just dying completely, the algae does die, but the chloroplast inside of that algae would remain. And now this larger eukaryotic cell just has the chloroplast. So you have some protists that have gotten photosynthesis in this way by engulfing algal cells and keeping their chloroplasts. So in this diagram you can see first the primary endosymbiotic events. Well, I guess they're actually not showing the very first endosymbiotic event in which eukaryotes acquire mitochondria, but this would be the second one, where you have a cyanobacterial cell being engulfed by an early eukaryote that already has mitochondria, and then this cyanobacterial cell is going to become the first chloroplast. So this early eukaryote with its chloroplast is going to differentiate into green algae and red algae, as time passes, and well, I should say its descendants would differentiate into the red and green algae, which are the true algae. Then you have some protists who engulfed those cells, either red algae cells or green algae cells, and got photosynthesis that way. So if you have a protist that engulfs a red algal cell, what's left behind is that chloroplast from that cell. You can tell it came from the red algae by looking at its pigments and by looking at its DNA. So you have a few protist lineages that have photosynthesis now because they engulfed a red algal cell in a secondary endosymbiotic event. We're going to go through all of these actually in this presentation briefly, but you have the Dinoflagellates, the Apicomplexans, and the Stramenopiles that all got red algae chloroplasts from a secondary endosymbiotic event in which their ancestors engulfed a red algal cell. Then for the green algae, you had a couple different lineages of protists engulf green algal cells and get chloroplasts from them. And again, you can tell those chloroplasts came from green algae by looking at their pigments and the DNA that they have. So today we have Euglenids and Chlorarachniophytes that have chloroplasts from green algae. So all of these guys over here are protists, but you could also consider them algae because they have photosynthesis, so it's a little bit confused. And any algae that's not either green or red could also be considered a protist. So you also have golden and brown algae, and those are also considered types of protists. Only the red and green algae are true algae. Every other microbial eukaryote that has photosynthesis has gotten it by engulfing a red or green algae in secondary endosymbiosis, or having been descended from, an ancestral cell who who had a secondary endosymbiotic event. So now we'll go over the major groupings of eukaryotes. The taxonomic groupings for eukaryotes are pretty messy. So normally we would think, well, for bacteria it goes from domain to phylum, then to class, order, family, genus, and then species. And then for eukaryotes, there should also be kingdom in there. So it should go domain, kingdom, phylum, class, order, family, genus, species. But as people have studied the phylogeny of eukaryotes, it turned out that that wasn't good enough, that you needed additional taxonomic groupings. So now we've added supergroups above the kingdom level. So it goes now domain, supergroup, kingdom, phylum, class, order, genus, species, class, order, family, genus, species. And even that was not enough. So we've got kind of super kingdoms, subkingdoms, infrakingdoms. Then we have superphyla, subphyla, infraphyla. So to be technical, it goes from supergroups to superkingdoms, then subkingdoms, then infrakingdoms, then to superphyla, then phyla, then subphyla, then infraphyla, and then to classes. So it's really complicated with a bunch of different taxonomic groupings at different levels. And it's, as with all phylogenies and all taxonomies, it's constantly being revised as more information is uncovered. So we're not going to worry about most of this stuff. We're just going to go through the 6 best characterized supergroups that you have in domain Eukarya, which are Archaeplastida, Rhizaria, Chromalveolata, Excavata, Amoebozoa, and Opisthocanta. Archaeplastida has the plants and the true algae, the red and the green algae. Opisthocanta has the animals and the fungi. Every other supergroup has protists. And the Chromalveolata has some protists that are also considered algae, which are the red, the brown and the golden algae. And that's just because of when they were discovered historically. They were not understood as protists, they were understood as algae, similar to how cyanobacteria were initially thought to be algae as well. So sometimes they're still called like blue-green algae, even though today we know that they're bacteria. Also for Chromalveolata, we are going to talk about 2 of the different superphyla within that supergroup, the Stramenopiles and the Alveolata. So in this diagram, you can see kind of a summary of a phylogenetic tree for eukaryotes. At the bottom, you have your early eukaryotes who gained mitochondria through a primary endosymbiotic event by engulfing a proteobacteria. Then on this branch here, which is for the Archaeplastida supergroup, you have the second primary endosymbiotic event in which a cyanobacteria was engulfed by an early eukaryote and became chloroplasts. So here you have your red and green algae and your plants. Then you have these dashed lines, which are showing secondary endosymbiotic events in which either a green algae or a red algae was engulfed by some protist and persisted in that lineage and gave rise to chloroplasts in those protists. So here you have the Rhizaria, which are all protists, the Chromalveolata, which are protists, but you also have the brown algae and golden algae here, which are protists but are also called algae. Just because of how they were discovered. They were initially thought to be algae. Then you have the Excavata, the Amoebozoa, and then finally Opisthocanta, the fungi and the animals. All the rest of this is protists. So we're going to start out by going through those 4 supergroups of protists. And we'll start with the Excavata. So you have actually a bunch of groups of protists in the supergroup Excavata. We're only going to worry about 4 of them: the Diplomonads, the Parabasalids, the Kinetoplastids, and the Euglenids. You do have a lot more than just that. And we're not going to worry about the exact taxonomic classification of these groups of protists because it's just so confused. The taxonomy is so confused and it's subject to change. So where these protists are classified exactly in the taxonomy might change, but the actual groups of protists, would not change. So starting with the Diplomonads and the Parabasalids, both of these groups of protists are flagellated, so they have flagella that allow them to move or swim around through liquid. They actually don't have mitochondria. They've lost their mitochondria, which makes them pretty unusual. They get their energy from fermentation, and they are anaerobic, so they only live in places where you don't have oxygen. A lot of them actually live in intestines of different animals. Some of them live there as parasites, so that they harm their animal host. Some of them live there as commensals, which would mean that they don't benefit or harm their host. They don't affect the host in any way. The Diplomonads can always be recognized by their double nuclei. The Parabasalids have something called a parabasal body. The function of the parabasal body actually is not known. We just know that it is next to the Golgi apparatus and seems to support it as part of its function. So here you have an image of some Diplomonads, and you can see these two kind of pale circles. Those are their two nuclei. And here's Parabasalids, and you actually are not able to see the parabasal bodies on these images. For the Diplomonads, you have one genus that's important for humans, Giardia, because it contains human pathogens and causes the disease giardiasis. Then for the Parabasalids, something that's unusual about them is that most of their genes don't have introns, so they wouldn't undergo RNA splicing like most genes do for eukaryotes. They're also unusual because they have really big genomes. Some of them have like some of the biggest genomes for any eukaryote, actually, even though they're just a single cell, so it's pretty unusual. You also have a genus Trichomonas that contains human pathogens that would cause the disease trichomoniasis. Then finally, we have the Kinetoplastids and the Euglenids. The Kinetoplastids all have the structure called the kinetoplast, which is actually just the DNA inside their mitochondrion. They would all have just one mitochondrion. So all mitochondria have DNA in them, but with the Kinetoplastids, the DNA is kind of like extra dense, so it's actually visible under the microscope, and we call it the kinetoplast. And you can see that in this image here of Kinetoplastids. Right here actually is the kinetoplast in both of these cells, and this would be the nucleus, and then the DNA in the mitochondrion is the kinetoplast. Most kinetoplastids live in the water and are predators that eat different types of bacteria, but there's some exceptions, and you have the genus Trypanosoma that has human pathogens in it. Those pathogens cause African sleeping sickness and Chagas disease. Chagas disease is actually something that we have around here. Those diseases are caused by kinetoplastids that actually infect insects, so the disease is transmitted by insect bites. Then you have the Euglenids, which are chemotrophs and phototrophs, so they can get energy from light and from chemicals. So since they're phototrophs, of course, they have chloroplasts, which they got through secondary endosymbiosis by engulfing an algal cell. They also usually live in the water, and they have flagella. So they'll use the flagella to kind of swim around in the water to find the spot that has the light that's best for them, either the best light levels or the best available of the chemicals that they're eating as nutrients. Some of them are also predators of bacteria as well. So here are a couple of images of Euglenids, and in these ones you can actually see that they're green from their chloroplasts. The next supergroup is Chromalveolata, which first we're going to talk about the phylum Alveolata for that group, and then next we'll talk about the other phylum, the Stramenopiles. Sorry, superphyla, not phyla. One thing that's special about the superphylum Alveolata is that all of them have a structure called the alveolus, or alveoli, it would be plural, which is a little sac just inside the membrane, and no one knows what it does, but they all have it. So there's a few different theories about what it does, but we just don't know for sure. Again, there's a lot of different groups of protists within the superphylum Alveolata. We're only going to worry about a few of them: the Ciliates, the Dinoflagellates, and the Apicomplexans. So first, the Ciliates, which are called that because they have cilia. A lot of them will actually have multiple life stages. So just one stage needs to have the cilia. For some of them, multiple stages have them, and for some of them, just one life stage will have the cilia. Cilia are those little hairs that can kind of cover the cell surface and move around, kind of beat. So they can be used to move the cell, or they could be used to move particles. So they're used for different things. For the ciliates, they are used both to move the cell and to move particles. Ciliates will usually have a particular spot on the cell that's serving as their mouth, and the cilia will frequently be used to move food particles in towards that mouth. So you have a couple images of ciliates here. In this image, you can see kind of the halo created by the cilia around the outside of the cell. And on this one, you can see the cilia really clearly. They look like all these little hairs. And here's actually the mouth. So a lot of these cilia would be acting to move food in towards the mouth. Ciliates are unusual because they have two nuclei, and they're not the same size. So one of them is the macronucleus, which is like the regular nucleus, and then the other one is the micronucleus, where you have specific types of genes. Most of the genes in the micronucleus are related to sexual reproduction. So the ciliates are able to do sexual reproduction, but it's not kind of like the normal type that we would think of as being sexual reproduction. When the ciliates are doing it, actually what they're doing is just switching their micronuclei off with each other, which does count, I guess, since they're sharing genes. You also have some ciliates that have endosymbionts living inside of them, or are symbiotes themselves. So they would be living inside of a host, for instance, in the intestines of an animal, either as mutualists or parasites. So either harming the host as a parasite or helping the host as a mutualist. An example would be in the rumen, which is part of the stomach that you have in cows and deer and just animals like that eat grass. Those animals aren't able to digest the grass themselves, but they have microbes in their rumen that are able to digest the grass. And so some of those microbes are actually ciliate protists. You have one famous genus in this grouping of protists, which would be Paramecium, just because it's really well studied. Then you have the Dinoflagellates, which are phototrophs and have chloroplasts, which they got in a secondary endosymbiosis event. They live in the water, as probably most protists do actually, and they also have flagella. Actually, some of them are living as part of coral reefs in a mutualistic symbiotic relationship. Some of them also can generate bioluminescence, and a few of them are toxic. So specifically, or most commonly, the genus Goniolix, which when it forms blooms because of overpopulation or a population explosion, then you have a lot of those toxins that it produces, and they can kill fish and poison people. So this is an example of a Goniolix cell, and here is a bloom caused by Goniolix. So this would be toxic. This is called a red tide. These types of blooms are called red tides, and you don't want to get in the water when you see the red tide. Next, you have the Apicomplexans, or finally, you have the Apicomplexans, which are obligate parasites, which means they don't have any other way of living than to be a parasite. If they try to live on their own without a host, they can't do it. They will die. They have multiple kind of life stages. As an adult, they are not motile. As a non-adult, they are motile. So if they're not in the kind of adult stage, then they would be called a sporozoite, which is basically the form they take when they're trying to switch from one host cell to another. So this sporozoite is really good at entering a host cell, and then once they're inside the host cell, they would turn into an adult and lose their ability to move around. So in the picture here, you have, this one is an adult apicomplexan, and these guys are the sporozoites that are designed to transfer between hosts. Apicomplexans all have a structure called the apicoplast. The apicoplast is actually related to chloroplast, but it doesn't do photosynthesis anymore. So kind of the ancestor of the apicomplexans would have gotten chloroplasts by secondary endosymbiosis, and then in the apicomplexans, those chloroplasts kind of lost the genes that they need in order to be able to do photosynthesis. So they don't do photosynthesis anymore, which means they're not really chloroplasts. Instead, they're apicoplasts. But they do stuff. They do some stuff. So they're synthesizing different things for the cell. For example, like free fatty acids and heme groups, which would be used by certain enzymes. It's like a group that can bind iron, so enzymes that need iron for a cofactor would also usually need a heme group to bind it. You have a couple genera that are important for humans, the Plasmodium and Toxoplasma genera, which both have human pathogens in them. Genus Plasmodium has the pathogen that causes malaria, and then genus Toxoplasma has the pathogen that causes toxoplasmosis. The other superphylum in supergroup Chromalveolata that we're going to talk about is the Stramenopiles. These all have flagella, but they're not like normal flagella. They've got kind of little structures coming off of them that look like little hairs, and they all have that. Some of the Stramenopiles actually aren't even microbial. Some of them are called macroorganisms, so they're like, the type of organisms that you can actually see with your eye. So we're not really going to worry about those, of course. The major groups that you have in the superphylum are the diatoms, the oomycetes, the golden algae, and the brown algae. And especially it's the algae that can be macroorganisms sometimes. You also have other groups beyond that, of course. Those are just the ones that we're going to talk about. And again, those golden and brown algae are protists. They're photosynthetic protists that have gotten chloroplasts as a result of secondary endosymbiosis. First, we have the diatoms and the oomycetes. The diatoms are actually also phototrophs, so they have chloroplasts that they got, through secondary endosymbiosis by engulfing an algal cell. What's really unique about the diatoms is their cell walls, which are made out of silica, which looks really similar to glass, or is pretty close to glass. So their cell wall can form all these crazy shapes, and can have a lot of detail on the cell wall, and just it's very beautiful. So they're kind of famous for having the prettiest cell walls of them all. They don't have flagella, so they're non-motile, but they do live in the water, so they just kind of float around. Actually, a lot of the plankton that you have in the oceans are just diatoms, so they're kind of the basis for a lot of food chains in the ocean, or I should say for oceanic food chains. And because their cell wall is made out of silica, which is very hardy and it doesn't degrade, whenever they die, the cell wall will form a frustule, which, a frustule is just the cell wall of a diatom that's left behind when it dies. It's kind of like a fossil of a diatom almost. And then those frustules will fall to the bottom of the ocean, and they're really useful, just to study, to understand kind of the history of diatoms in the ocean. Then you have the oomycetes, which look really similar to fungi, actually. So they have filaments, and there's not really divisions between the cells in the filaments, which gives you like filaments that have like multiple nuclei in them, which is just like a lot of fungi have. A lot of fungi also, like, they form filaments, and then they don't have divisions between the cells and the filaments, so they all kind of are like merged together basically, and you'll have multiple nuclei in those filaments. So because they look so much like fungi, they actually used to be considered fungi, and they're called water molds. So back whenever we thought that they were fungi, it was the group of fungi called the water molds. And so that name has stuck around even though today we know that they are protists, not fungi. So here you can see an oomycete with the filaments. You can't really see the nuclei in there. You can see little spores that are formed at the ends of some of these filaments, but there's not really divisions between the cells in these filaments, so you would have multiple nuclei inside of them. So since they're called water molds, you might figure they grow in the water, and they do. And specifically, they would live on decaying organic matter in the water, like dead fish or dead plants, stuff like that. We do have the one genus though that's really important, or has been very important for people, which is Phytophthora. So you guys have probably heard of the potato blight in Ireland, or the potato famine, where the main crop for Ireland was the potato, and all the potatoes died one year because of a potato blight, which is actually caused by an oomycete protist in the genus Phytophthora. And a lot of people actually starved because there was no food. All the potatoes had died. Next we have the golden and brown algae, which are protists even though they're called algae, but you could also think of them as algae because they're phototrophs that have chloroplasts, which they got from secondary endosymbiosis. So they all live in water, usually the ocean, and they actually have a special pigment, which is chlorophyll C. It's actually the same pigment that the diatoms have. So your plants and your red or green algae, which are the true algae, have either chlorophyll A or chlorophyll B, or they have chlorophyll A and chlorophyll B, but they don't have chlorophyll C. So you had a secondary endosymbiotic event where the ancestor of these golden brown algae and the diatoms, engulfed an algal cell, and the chloroplast of that cell remained in the lineage, and as time passed, the pigment kind of evolved, or the enzymes that create that pigment evolved. So instead of creating chlorophyll A or B, they started creating chlorophyll C. So all these golden and brown algae in the diatoms now have the chlorophyll C. So that lets you know that even though these are called algae, they are not directly related to the red and green algae, since they have that different pigment. The golden algae are unicellular in general, whereas the brown algae are multicellular. So a lot of the brown algae actually are macroscopic, and you can see them with your eyes. They are not microbes. For the golden algae, most of them are phototrophs, but actually some of them have lost photosynthesis, and they're now living as predators on bacteria. For the brown algae, basically kelps and most seaweeds that you would think of are actually algae. So there are different types of brown algae. So here you have kelp, that's a type of brown algae, a macroscopic organism, of course. And then you have a couple examples of the golden algae, which would be unicellular. Our next supergroup is Rhizaria. One thing the Rhizaria protists all have in common is they have pseudopodia, which are structures that are like little fingers basically sticking out from the cell membrane, or I guess it's actually the cell membrane itself sticking out to make like a little finger or a little thread going out. So the pseudopods are used to move the cell around and also to help it eat. They actually function similar to how amoeba would move around, which we will go over when we talk about the amoebozoa. But they kind of stick out a pseudopod, it attaches to the surface, and then the cell will kind of pull itself up to that pseudopod and then stick out a new pseudopod, further ahead. So you have a lot of groups of protists in the supergroup Rhizaria, but the ones that we're going to talk about are just the Chlorarachniophytes, and the Foraminiferans, and the Radiolarians. And of course you have more beyond that we're not going to talk about. So first we have the Chlorarachniophytes, which are also phototrophs that have chloroplasts that they got from secondary endosymbiosis. In this case, they engulfed, or you had, an ancestor of a Chlorarachniophyte that engulfed a green algal cell. And normally in secondary endosymbiosis, that algal cell would degrade and all you have left is the chloroplast of it. But in this case, the algal cell never fully degraded, so you have more parts of it left behind than what's normal. So Chlorarachniophytes have really unusual chloroplasts because of that. They actually have 5 membranes in total, and they have a structure in them called the nucleomorph, which is basically the remainder of the nucleus from that green algal cell that they engulfed, or that their ancestors engulfed so long ago. So you have these 2 membranes around the outside. So there's one in kind of this like brown line, that's one membrane. And then you have the blue line, that's another membrane around the chloroplast. Then inside of there, you have the nucleomorph. And then you have additional membranes. So this kind of light green line is a membrane. And then where this dark green starts, that's another membrane. So that's 4 membranes, and then of course you just have the membrane, of the thylakoids on the inside of that chloroplast. So that's the 5th membrane. So, one of these outer membranes is actually from the algal cell, the green algal cell, and the nucleomorph is from the algal cell. And then this outermost membrane would be from the host. When that host cell engulfed the algal cell, it would have encased it in a vesicle, and that membrane, is what that is. Like so many of these protists do, the Chlorarachniophytes also live in the water. Then you have the Foraminiferans and the Radiolarians. Both of these groups of protists have structures called tests, which is basically like a shell for the protist. So it's a structure made out of mineralized calcium that surrounds the protist and kind of protects it. So you can see examples of tests here. This is a foraminiferan test, and this is a radiolarian test. So you can see, like, kind of similar to the diatom shells. These are really complicated structures that can be very beautiful. But this is actually not a cell wall. So inside of the test, you would also have, like, the normal cell wall. Both of these groups also live in the ocean, as so many protists do, where they feed off of decaying organic matter, like fish or plants or algae. The foraminiferans actually will usually live deeper in the ocean, pretty much right by the bottom of the ocean, because their tests are usually like big and heavy. So you can see like the foraminiferan test is much bulkier here than the radiolarian test. Because of that extra bulk, they kind of sink to the bottom of the ocean. They're not able to float well. Some of the ones that don't live like right at the bottom, actually, that would have like lighter tests, so they're able to float somewhat, will actually have endosymbiotic algae living inside of them and providing them with photosynthesis somewhat. Then the radiolarians will live closer to the surface, so their tests are not as heavy, which makes them much better at floating. But they also have a lot of lipids and vacuoles inside of them. Like vacuoles being like little, not little, but large membrane sacs that they fill up with like gases that will help them float, so that they can float even higher than they would just based on the weight of their test. They would also a lot of the time have symbiotic relationships with algae, but just not like endosymbiotic relationships. So the algae would not be actually inside of the radiolarian, it would be like outside of it. The last supergroup of protists that we'll talk about is the amoebozoa, which also have pseudopods similar to the Rhizaria. The difference is that the pseudopods of amoebozoa or for amoeba are like bigger and thicker than in Rhizaria, which are like really narrow, thin pseudopods. But the function is the same, so they're used to move the cell around and to help the cell eat. You have, again, as always, a lot of different groups of protists in the amoebozoa, but we're only going to talk about 4 of them: the Gymnamoebas, the Entamoebas, the plasmodial slime molds, and the cellular slime molds. So first, the Gymnamoebas and the Entamoebas. which are both types of amoebas. The Gymnamoebas live in the water and in the soil. They're mostly predators that eat bacteria or other protists, but some of them are more like scavengers that would eat decaying organic matter. They all have their pseudopods, of course, so they move using something called amoeboid movement, which is where they stick out a pseudopod and it attaches to a surface, and then the whole cell kind of moves towards that pseudopod, and then they stick out a new pseudopod. So here you actually have kind of a time lapse of that amoeboid movement. So this is a pseudopod that this Gymnamoeba is sticking out. Here it is, fully extended. Then the cell is going to kind of move over in the direction of the pseudopod, which brings it forward. And then it can start sticking out a new pseudopod. So when the cell is actually kind of moving towards like the pseudopod that it's actually stuck out, or that it's just stuck out, it's kind of a little weird how that happens, but it's like the cytoplasm, the liquid of the cytoplasm is like flowing down into that pseudopod, and as the cytoplasm is flowing into the pseudopod, it kind of carries like the body of the cell with it. I mean, I guess, of course, the cytoplasm is all like the stuff inside the cell except for the nucleus, but it's going to like carry the cell membrane with it so that the whole cell will move, towards that pseudopod. So here you have kind of an example of the cytoplasmic flow. It's also called cytoplasmic streaming. So you can have, under normal conditions, the cytoplasm just moving around kind of randomly in or out of different extensions of the membrane, which is not going to lead to, movement really. But if you have the whole cell kind of straighten out, and you're sticking a pseudopod out on one end, then the whole cytoplasm can move like down into that pseudopod, and take basically the cell membrane with it. Then you have the Entamoebas, which also use pseudopods, but not as much for movement because they just don't need to move as much since they live inside of hosts. They're not predators like the Gymnamoebas are. So they're parasites living inside of some host, which is usually an animal. And we have one genus in there that's important for people, which is the genus Entamoeba, that contains human pathogens, causes a disease called amoebic dysentery. And finally, we have the slime molds, which includes the plasmodial slime molds and the cellular slime molds. Both of those groups used to be considered fungi, which is why they have the name slime mold. And again, just like the oomycetes, it's because of the way they're shaped. They look like fungi. So they have, filaments that, sometimes will have like no boundary between the cells, so it's like there's multiple nuclei, or else there are boundaries, but it's still like kind of filamentous. Slime molds are actually really quick moving. They also use the pseudopods and amoeboid movement, and they move like surprisingly fast. And they can move kind of all as a group. So the whole like mold will move like together. And, it can be visible to the eye. So each individual cell, is microscopic, but when they get together in these large groups, you can see it and it looks like a mold. You find them living in the soil, also on decaying organic matter. Some of them are also predators, though, that would eat different bacterial species. When they're ready to kind of reproduce, which they are actually able to reproduce sexually, so whether they're reproducing sexually or asexually, they're going to form spores that would then kind of, be dispersed like farther afield in the environment. The main difference between the plasmodial slime molds and the cellular slime molds is that the plasmodial slime molds are not like divided into cells, so they're more like the oomycetes, where you don't have divisions between cells, which means you end up with these filaments that look like, they have multiple nuclei in them, and we call those plasmodia in the case of the plasmodial slime molds. For the cellular slime molds, the cells are all divided from each other, so it's clear there's one cell, one nucleus, but they also like get together in these large masses that would form the actual slime mold. On top here you have an image of a plasmodial slime mold. So if you zoomed in here, you would see like that these filaments don't actually have like divisions between the cells in them. And this is like the life cycle for a cellular slime mold. So, you know, those cells are going to get together and form these kind of large structures. Then if you zoom out, it looks kind of like this. So fungus-like. Then here you have some pseudopods and like the little trails that they've left as they're moving around on the agar. This is a fruiting body that they form as they're going to reproduce, and then they make these spores, and then here's like a zoomed out picture of the spores. So next we'll go over Opisthokonta, which is a supergroup that has the fungi in it. It also has microscopic worms, but we're just not really going to talk about them. We'll just talk about the fungi. So, about the fungi. Most fungi are microscopic, but you also have fungi that are macroscopic, so you can see them with your eye. They live in the soil usually, or on decaying organics. A lot of them are multicellular. Well, most of them are multicellular, but you also have some that are unicellular, and you have some that could be unicellular or multicellular, depending on the conditions, like of their environments. All of them can at least reproduce asexually, but a lot of them can also reproduce sexually as well. Again, which one they do, sexual or asexual reproduction, would depend on their environment and the conditions of their environment. All fungi will have cell walls that are made out of chitin. Chitin is a polysaccharide that's actually kind of similar to cellulose. It's really strong, and for animals like shrimp shells and stuff, like crustaceans, those shells will be made out of chitin as well. Most fungi are not harmful to humans, so they don't cause disease in humans. You do have a good number of fungi that are pathogenic to plants. So, you know, for agriculture, you know, fungi end up being more important because a lot of them do cause like plant diseases, but they usually don't cause disease in people, which is partly because the human body is just a little bit too hot for most fungi to be happy living there. When you have a fungus that does cause disease in a person, it's called a mycosis, or plural would be mycoses. Most healthy people are not vulnerable to mycoses, or if you do get a mycosis, it's going to be kind of minor, and you know, it'll just stay like wherever it started at. For people who get like seriously sick with mycoses, usually they have something wrong with their immune system. So they're immunocompromised for some reason or another. There's a lot of reasons why you might be immunocompromised. So kind of the most famous reason would be just if you have HIV, you know, that the HIV virus destroys the immune system, and then you end up with AIDS. And are quite vulnerable to a lot of diseases that would not be serious for the average person, including a lot of fungal diseases. But you also have other diseases that can suppress the immune system or just damage body organs, especially the lungs. A lot of the time, if you're going to get sick with a fungus, it's because you breathed in the spores, and the spores went into your lungs. So if you have some lung condition that has weakened your lungs, you're more vulnerable to a mycosis that would be transmitted that way by breathing in the spores. So something like cystic fibrosis, you know, damages your lungs and makes you vulnerable to lung infections, including like mycoses. Also, people who have to take immunosuppressant drugs, maybe they have an autoimmune condition, or maybe they had an organ transplant, and then they need to be on these immunosuppressant drugs, you know, to make sure that their immune system does not attack their own body. Those people are also immunocompromised and will be vulnerable to fungal diseases. Then for a lot of the mycoses that do cause disease in healthy people, usually they are breathed in through the lungs, and you know, so you breathe in the spores, and the spores kind of grow into a fungus in your lungs. For people with like a healthy, intact immune system, usually it's not going to get that serious, and it's going to stay there. For people who are immunocompromised and they breathe something in that causes a mycosis that initially starts in the lungs, it can get very serious. You can get like a big old ball of fungus like living in your lung, and a bunch of destroyed lung tissue that's been replaced like with this fungus. But also the fungus is more likely to spread to other parts of your body, and ultimately kill you. But for the average person, it's not that dangerous. Oh, well, I guess I should also mention that some fungi would make toxins so that if you ate them, you might get sick. But it's not like a pathogen where the fungus is living inside of you and making you sick. It's just that it's created this byproduct that you ate that's toxic for you. A lot of fungi are actually symbionts of plants, so they're not just hurting the plants, but they could be helping the plants. Like most plants actually will live with some type of fungus associated with their root system to help them absorb nutrients as part of like a mutualistic symbiotic relationship where both the plant and the fungus are benefiting. All of the fungi are chemoorganotrophs. So you don't have any phototrophs in here, there's no chemolithotrophs in here, just chemoorganotrophs. They always are getting their energy and actually their carbon too from some organic source. So they're kind of, they're scavengers for the most part that would, eat decaying things. In general, the way that they eat is by secreting enzymes onto whatever they're going to be eating. Usually it's something that's dead, some type of dead organic material. They secrete enzymes onto it that kind of digest it, and then they suck up, the results, the digested material. Some of them would actually be doing that to plants or animals that are not dead yet. Fungi are really important for the ecology of the earth because they do eat decaying things. Of course, a lot of bacteria will do that as well, and insects actually do that too. So kind of between the bacteria, the fungi, and the insects, you have, well, and I'm sure archaea in there as well, we just haven't studied them as well, you have the ability to decompose dead things. So if you didn't have those types of organisms, then when something dies, it would just stay there forever, basically, and the world would be, just one big mound of corpses. So we're really indebted to the bacteria, to the fungi, to the insects for breaking down this dead stuff and getting rid of it for us, so new things can come in and grow. For fungi, one thing that's special about their ability to decompose dead stuff is that a lot of them can decompose wood. Wood is really hardy. It's really hard to degrade wood, and a lot of kind of insects and bacteria that are scavengers feeding off decaying stuff. They're not eating wood. They can't do that. Most cannot do that, but a lot of fungi can. So first we'll go over the general morphology of a fungus, or the general shape of the fungus and the different structures that they have on average. Of course, there's specific fungi that would have different structures, instead of or in addition to these structures. This is just the basics. So in general, if you have a single-celled fungus, that's called a yeast. If you have a multi-celled fungus, that's called a mold. For the yeast, the single-celled fungi, they're going to reproduce asexually using budding division. Some of them can reproduce sexually as well, but they can all reproduce asexually by budding. For the multi-celled fungi, the molds, they're going to form these kind of masses of filaments. Each individual filament is called a hypha, or plural hyphae. Then the mass itself is the mycelium, or if you had multiple masses, it would be mycelia. So a mold itself is like a mycelium, or like the body of the mold is the mycelium, and then the individual filaments are the hyphae. For any mold, the hyphae could be septate or they could be aseptate. If they're septate, it means that there's cell walls that separate the individual cells, so you can actually see the individual cells, and each one has its own nucleus. For the aseptate hyphae, you don't have those cell walls, dividing cells in the filament. So it's just continuous, and you have multiple nuclei, in that filament. So that's more like the oomycete or like a plasmodial slime mold. And then the super fancy word for the aseptate hyphae is sonocytic. Actually, I'm sorry, that's cynocytic. So you spell it Coenocytic, but you say it cynocytic. So any fungus is going to be like if it's a mold, any mold fungus is going to have a mycelium, is going to have a hyphae that are either septate or aseptate. And then some of them might have additional structures on top of that. That would be for usually for reproduction. If you have a mold that's going to reproduce asexually, usually you're going to have hyphae that come up from the mycelium, and you have spores produced on the ends of them. And those spores are called conidia. So conidia are for asexual reproduction. Then if the fungus is going to reproduce sexually, it's going to make a different type of spore on like a different structure. And what type exactly or what structure it makes exactly to hold those spores is going to depend on the specific type of fungus. So for fungi that make mushrooms, those structures are actually to hold and distribute spores for sexual reproduction, not for asexual reproduction. So then we have some images here. So this is actually the conidia. So each of these little balls is a conidiospore. This is the hypha that's sticking up from the rest of the mycelium that has them on it. So in this diagram you can see that kind of better, where this whole thing would be the mycelium. These are the hyphae, and then this hypha is sticking up vertically and is forming the conidia on the end of it. So each of these spores would be able, to turn into a new mycelium. At the bottom here you have the septate hyphae versus the aseptate hyphae, or the cynocytic hyphae. So in the septate you can see the cell walls that divide one cell from the next cell, so that each cell has, is clearly visible where it begins and ends, and it has its own nucleus. In the aseptate or cynocytic hyphae, you don't have those divisions between cells, so you still have the nuclei, but you can't see where one cell ends and the next begins, so it's all just kind of one mass. Then this is an image of a mold growing on a petri dish, and you can see it looks a bit fuzzy. It's fuzzy because of all these conidia and these special hyphae that are coming up and have conidia on the ends. And you can't see each individual one, but you can see the overall fuzzy appearance. And then this is, yeast. So these are yeast cells, and you can see some of them budding. So all these cells that have like a little thing sticking off of it, those are cells that are in the midst or in the middle of budding, reproducing by budding. Next we'll go over briefly symbioses that fungi will commonly form. First, the symbioses they form with plant roots. Most plants do prefer to live in a symbiotic relationship where they have, fungi on their roots, helping them absorb stuff from the soil. And usually it's like specific nutrients that they absorb better when they have the fungus living with them. A common one is phosphate. And then what the fungi are getting is, they're getting nutrients back from the plant roots. So the plant is getting nutrients in the form of like minerals from the soil that the fungus is helping them absorb, and then the fungus is getting nutrients from the plant like sugars and stuff that the plant is producing using photosynthesis. And you have a special structure called the mycorrhizae. Actually, that's a plural form of the word. So mycorrhizae mycorrhizae are structures on these plant roots that are formed by the fungus living there symbiotically. And you have a couple of types. You have the ectomycorrhizae and the endomycorrhizae. The ectomycorrhizae are made between a specific type of fungus called the Basidiomycete and plants that are woody. So, they have like a woody stem or a trunk. The endomycorrhizae are formed between a type of fungus called glomeromycete and a non-woody plant. So, just one that doesn't have wood, doesn't form wood. For the endomycorrhizae, the endo means inside of, so the hyphae of that fungus are actually penetrating into the plant cells to kind of deliver nutrients into the plant and to take up like sugars from the plant more better. For the ectomycorrhizae, ecto means on top of, so in that case the fungus is just on top of the root and it's not actually going like inside of it. So here you can see an example of like a root ball with the mycorrhizae on it. All this fuzzy stuff, that's fungus that's helping the plant absorb nutrients. You can actually buy like fungus to add to your plants, add to your like potted plants to help them grow. That would basically serve as mycorrhizae. And you know, plants are just a lot more efficient at taking up nutrients from the soil if they have those mycorrhizae with them, for most plants. Then the other common symbiosis for a fungus to form would be a lichen. A lichen is like a composite organism where you have a fungus in there and you're also going to have something else. It could be algae or it could be cyanobacteria, so usually either the cyanobacteria or the algae. You know, and they're living together. The fungus is kind of providing like a structure, and then the cyanobacteria or the algae is providing photosynthesis. So here you have a couple different types of lichens growing on, looks like a rock. So this pale green one, that's one type, and then this kind of leafy, brighter green one is a different type. There's a lot of different types of lichens. And finally, before we get into like the phylogeny and taxonomy of the mushroom or of fungi, we'll go over the general kind of spores that are used for sexual reproduction in fungi. So different types of fungi will have different types of spores that they use for sexual reproduction. Then they'll also have different structures that they make to house those spores and support them. And the kind of the type of spore that each fungus has for sexual reproduction and the structures that are formed to house it and stuff is actually used to classify those mushrooms into different groups. So first you have the ascospores, which are, you know, all what they have in common, I guess, is that they're all formed in this little sac. It's called an ascus that protects them. Actually, a lot of the time you have yeasts that will form ascospores. So if yeasts are going to reproduce asexually, they'll do it by budding. If they're going to reproduce sexually, a lot of them will actually make ascospores. And you can see some ascospores right here. So each of these kind of darker, like black or brown balls is an ascospore. And it's hard to see, but there's actually an ascus that each of these lines of spores is contained inside of. So right here you can kind of see it like this long kind of finger looking thing. This is the ascus, and that would be full with the ascospores. So each one of these lines of spores is actually being contained inside of one of those asci. Then you have basidiospores, which are formed on something called a basidium, which is usually found on the bottom of a mushroom. So here you have a mushroom. The whole mushroom, the cap of the mushroom when it's mature is actually called the basidiocarp, and then underneath it you would have the basidia, and each one would house some basidiospores, and then those spores here are being released and germinating to form a new mycelium that eventually makes a new mushroom. Then you have zygospores, which are shown right here. These are formed in an unusual way, so you would have like a couple different hyphae kind of fused together to form these spores, and then the dark balls are the actual zygospores. So then these kind of like branches almost leading up to them would be the hyphae that fuse together to form the zygospore. And then you have at last the zoospores, which actually have flagella on them. So this is like a, what do you call it? Immunofluorescence, right? This is a fluorescent stain, so you can actually see the glowing stain. And the zoospore itself is stained in red, and then the flagella coming off is stained in green. So each of these spores would be formed by a specific type of fungus, and we'll go through those different groups in a moment. So here's a basic overview of the phylogeny for fungi. You have 9 different phyla of fungi, but sometimes they call them divisions instead of phyla. You know, they just like to do it a little different for fungi. So sometimes you might see these names referred to as divisions, like division Ascomycota or phylum Ascomycota. It's basically the same thing. The phyla are, well, the taxonomy and the phylogeny for the fungi is constantly being changed and updated. So the 6 phyla that you have in this diagram on the side here, these are the 6 phyla that we're going to talk about today, but actually one of these, the Zygomycota, that's not a phylum anymore. It got split up into 2 different phyla. And a similar thing may happen for some of these other phyla, where you have some species in the phylum that we kind of know that they don't go there. They are there for now, but we know that, you know, there's something not right about that phylogeny. So, you know, for some cases, you might have some species that get pulled out of one phylum and, you know, replaced into another, or it might be a thing that happened with the Zygomycota again, where the whole phylum gets split up into multiple different phyla. So out of all these different phyla, well, all the 9 total that we have so far right now, most of the fungi that we actually know something about are belonging to just 2 of the phyla, either Ascomycota right here or Basidiomycota, which are the ones that make mushrooms. Well, most mushrooms actually are in Basidiomycota. So most of the fungi that are out there, we really don't know that much about them and haven't characterized that many different species of them. And of course, since the fungi are together with the animals in supergroup Opus thecanta, out of all the other groups of eukaryotes, fungi are most closely related to the animals. So we'll start with the phylum Microsporidia. So it's not really clear if these are even fungi. It used to be that the Microsporidia were considered to be protists. Now we know that they're obviously really closely related to fungi, but we're still not sure if they are fungi. So they are classified as fungi right now, but they might be moved somewhere else in the future. This type of thing is really why you end up with supergroups and so many, weird groupings for the eukaryotes. It's like a fungus, but it's, not quite a fungus, but we don't know what else it would be, So we're probably going to just make up a supergroup or, a super kingdom for Microsporidia at some point, maybe. But for right now, we're calling them fungi. All of the Microsporidia are obligate parasites, so they cannot live without a host. They're either going to be parasites of animals or of protists. They don't have mitochondria, they don't have a Golgi apparatus, so that's partly the reason why they're stuck as obligate parasites. Since they don't have mitochondria, they don't really have a way to make a lot of energy for themselves, and they're going to steal it from their host. Some of these are human pathogens, as you might figure, since they're obligate parasites. Usually they just affect immunocompromised people though, so people with a healthy immune system wouldn't really be affected by them. But in people who are immunocompromised, they can cause serious conditions, well, specifically microsporidiosis. In the images here on the bottom, you can see some cells that are infected by Microsporidia. So all these small dark dots are Microsporidia. The large blue dots are nuclei of these larger cells that are the host cells for these Microsporidia. And then here you have a life cycle diagram for a couple common species that cause microsporidiosis in immunocompromised people especially. So these are the spores over here. They're going to find a host cell. Then they inject the host cell with the contents of the spore. So everything inside the spore gets injected into the host cell. And then ultimately it's going to form new spores that when they're mature will kind of burst out of the host cell and start the cycle over. And then each of these red dots is a part of the body where it would be common to have, a microsporidiosis infection if you're immunocompromised. Oh, in the lungs, of course. Next, we have phylum Chytridiomycota, which is a very ancient phylum. So out of all the phyla of fungi, this is probably the oldest one. And it's also one of the ones where we know that some of the species in this phylum actually should go somewhere else. We just don't know where else to put them yet. Every fungus that is a part of this phylum is called a chytrid, and they all reproduce sexually using the zoospores, which were those flagellated spores, so they can move around. The chytrids will live usually in water or in wet soil, so they need moisture of some kind. Some of them have just one cell, some of them have multiple cells. Some of them are like scavengers that decompose, organic matter. Some of them are parasites that, live on a host. And some of them are parasites of amphibians. So you might have heard about like the recent, well, I guess by now it's been going on for quite a while, but there's been a lot of mass amphibian deaths throughout the world. Where just, the frogs are dying, you guys. And it's actually a chytrid fungus or a chytrid fungus that's responsible. Well, that plus climate change, which is making, the world warmer. And so it's basically making a nicer environment for these chytrids, and then they're spreading beyond their kind of usual range and reproducing at an increased rate. And these ones that are specifically parasitic to amphibians are killing massive numbers of amphibians. So here you have a section of the epidermis of the frog, so just frog skin. And on top of it, you have these chytrid fungal cells that are parasitizing this frog. Or I should say were, since it's all sectioned and it's all dead now. Then we have the two phyla Zygomycota and Glomeromycota. These both have aseptate hyphae, also known as synocytic hyphae, so no divisions between the cells in their hyphae. The Zygomycota is a former phylum, so now all the species that were in Zygomycota have been divided up into two new phylums that it kind of was split up into. But we'll still talk about them together as though they did not belong to two different phyla now. So species in the former phylum, Zygomycota, live in the soil. They live on decaying plant matter a lot. Actually, some of them are bread molds, especially the fungi in the genus Rhizopus are bread molds. And that's what you can see here, a bread mold. And if you zoom in, you can see the zygospores. So fungi in this former phylum all reproduce in a similar way. They have kind of a cycle that they follow, where first they do a round of asexual reproduction, then they do a round of sexual reproduction, and when they're doing their sexual reproduction round, they're forming the zygospores, which are the ones formed by like two hyphae fusing together. You also have some human pathogens in this former phylum. Then for the Glomeromycota, those do not include pathogens. Those are symbiotes that live, with plant roots in the endomycorrhizae. So they would be forming this symbiotic relationship with non-woody plants and sending their hyphae actually into the plant cells to help deliver nutrients to them and also accept nutrients from them. They are obligate symbionts, which means they cannot survive without their plant partner. So the plant needs them, and they need that plant. They're not known to reproduce sexually, only asexually. And so you can see them down here. These like kind of thick brown lines, these are plant roots. And then the kind of, it looks a little fuzzy, kind of a little white, little fuzzy. That's all the Glomeromycota fungus forming endomycorrhizae with this plant. And then you can see the little brown balls. Those are spores like this. Asexual spores, so they'd be conidia. They aren't formed like kind of in the normal structure where you have a hyphae sticking up and conidia at the end. because these fungi live underground. Then we have the phylum Ascomycota, which is one of the ones that contains a lot of different fungal species that have been characterized, so it's one of the better understood phyla. All the fungi in this phylum are called Ascomycetes, and they all reproduce sexually using the ascospores, which would be formed in the ascus or asci sacs. You have a bunch of different genera in here that are useful for us or important to us for one reason or another. So first you have the genus Saccharomyces, which includes yeast, baker's yeast that you would use to bake bread. So here you have images of the Saccharomyces yeast budding, so reproducing by budding division. The specific species that is baker's yeast is Saccharomyces cerevisiae. So you can also use that to make beer. And then here you have a yeast that is in the process of budding, and on this side you can see like these little rings. Those are scars left by previous budding events. So this yeast has already done it's already budded like 3 times at least, and now it's budding a 4th time. Then you have the genus Candida, which is also yeasts. Some of those species in genus Candida are found living in the microbiome of the human body, especially Candida albicans. So you would have that in your mouth microbiome and also in the vaginal microbiome. It can form infections actually. So those species are pathobionts sometimes. Where it's fine to have them there, but if their population gets out of control and it's growing too large, then it can cause a problem for you, some type of infection. So for people who have AIDS or are immunocompromised, it's more common for them to end up with problems in their mouth related to Candida overgrowth. And also yeast infections that's caused by Candida overgrowth in the vagina. So if you were taking maybe a broad spectrum antibiotic and that kind of, decimated the population of bacteria in your microbiome, that's not going to affect Candida because it's a fungus. So a drug that kills bacteria isn't going to kill a fungus. So then now that yeast doesn't have anyone to compete with really, and its population is able to really explode, and then you have a yeast infection. Then you have the genus Aspergillus, which contains human pathogens. Usually they affect just immunocompromised people, but they can affect people with an intact immune system as well. Here's images of the Aspergillus and their ascospores. So all these little dots are the ascospores. So this is one of those fungi that would infect you if you breathe the spores into your lungs. And if you're immunocompromised, it can really form a large mass in your lungs and can migrate elsewhere in your body as well. If you have a healthy immune system, it's not likely to be serious, but that doesn't mean that it's impossible. Then you have the genus Penicillium, which is actually where we got penicillin from. So it was a fungus in the genus Penicillium that was the source of the first antibiotic. And of course, penicillin is not as useful as it used to be. Since it was the first antibiotic, now there's a lot of different bacteria that are resistant to penicillin. But, it is useful sometimes still. And it's historically important. And then you also have some edible fungi in phylum Ascomycota, specifically the morels and the truffles. Those are Ascomycetes. So here you have genus Morchella, which forms the morels. So that's it looks like a mushroom. It's technically not classified as a mushroom. Those would be, within those mushroom-looking thing, you would have the asci that are producing ascospores. And finally, phylum Basidiomycota, which is the other phylum that we know a lot about and have characterized a lot of the species that belong to it. Any fungus in this phylum is called a basidiomycete, and they all reproduce sexually using basidiospores. A lot of them will make mushrooms. Not all of them make mushrooms. They all make basidiospores, but they don't all make mushrooms. But a lot of them do make mushrooms. And the fancy name for the mushroom cap is the basidiocarp. So if you were to look on the underside of that mushroom cap, you have all these gills under there, and if you zoomed in on the gills, you would find these types of structures, where here you have a basidium, which is the base, that's used to produce these basidiospores. So multiple more than one basidium would be basidia. So these gills are lined with basidia, and they're all making basidiospores. A lot of Basidiomycetes are edible. A lot of different mushrooms are edible, but a lot of them are also toxic. So they're not infectious. Most Basidiomycetes are not like pathogens for humans, but a good number of them do produce, toxins. And then if you eat that mushroom, now you're eating the toxins. Some of them might just make you kind of sick. Some of them can actually kill you. And then you have this one genus, the genus Amanita, that has a lot of toxic mushrooms, including the death cap and the destroying angel. The death cap is actually the mushroom that has killed more people than any other mushroom. It's very toxic, and it kind of creeps up on you. So you eat it and then you get sick, and they say it tastes good actually. So it tastes real good. You eat it all, then you get sick, and then you get better, and you feel like, well, it's good now. And then a couple of days later, your liver fails and you die. So it's important not to just be eating mushrooms, because they can kill you, some of them. This mushroom over here is actually Amanita muscaria, also called the fly agaric mushroom. It's very distinctive because it looks so pretty. This is sometimes considered a toxic mushroom, but actually some people also just eat it. But, you shouldn't, if you see something like this, don't just eat it. There's specific ways that you're supposed to prepare it to reduce the toxicity so that it's safe to eat. Then you also have some mushrooms that are medicinal or hallucinogenic. Actually, the Amanita muscaria is a hallucinogenic mushroom as well. But most hallucinogenic mushrooms are in the genus Psilocybe. So Psilocybe cubensis and stuff like that, those are your magic mushrooms. And not only are they hallucinogenic, but they're also medicinal as well. They're very useful for treating cluster headaches, and like mental issues like depression, and also addiction actually. So that's not really prevalent in the U.S. It's certainly not FDA approved, but it does work and it is studied. And in other parts of the world, they do use psilocybe mushrooms as a treatment for addiction, and it does work. You also have a number of plant pathogens in this phylum. You may have heard of plant diseases referred to as smuts or rusts. That's like a type of plant disease. So any type of smut disease or rust disease, those are all caused by Basidiomycetes. And then finally, you have the genus Cryptococcus that has human pathogens that can cause meningitis. Again, usually just in immunocompromised people. Actually, those fungi in Cryptococcus can infect people with an intact immune system as well, but they would usually just stay in the lungs and not move to the brain, where they could cause meningitis. And finally, just a note on how mushrooms do look the same, and you should not eat them just because you found them and you think you know what they are. So in the corner here, we have Amanita bisporigera, which actually usually is like kind of a generic white-looking mushroom, but this particular instance of it, or this particular individual, is looking basically like the Amanita muscaria. But it's much, much more toxic. This is also sometimes called a destroying angel mushroom. So sometimes mushrooms will look different depending on kind of their growing conditions. So you can't always figure that you're gonna recognize them. And the fly agaric is like a really easily recognizable mushroom, of course. So you might figure that if you found something that looks like that, you know what it is. But it might be this guy as well. who's very toxic. So you can't just make that assumption. You would need to be a mycologist who, is someone who's trained to recognize different mushrooms, so that you could actually have confidence that you know what you're about to eat. Here you have Psilocybe cubensis, the magic mushroom. Here you have Amanita ocreata, which is the destroying angel mushroom. So sometimes the Amanita ocreata is called destroying angel, sometimes Amanita bisporigera is called destroying angel. Both of them are very toxic. And then the Amanita phylloides, the death cap mushroom that has killed more people than any other mushroom. And you can see like all three of these mushrooms, really, they look about the same. So don't assume that you know what it is because you found it on the ground. Don't eat it. And finally, we have the supergroup Archaeplastida, which has the plants and the algae, but of course, we're only going to talk about the algae. So first, we'll talk about the red algae. Another name for red algae is a rhodophyte. Most red algae live in the ocean, but some live in like terrestrial environments or in like fresh water. They could either be single-celled or multicelled, but most of them are multicellular. Some of them are macroscopic. Most of them are microscopic multicelled, but some of them are multicelled microscopic. And some of the seaweeds are actually red algae. A lot of them can reproduce sexually, but not all of them. And of course, since they're algae, they are phototrophs that have chloroplasts that do photosynthesis. And the pigments they have are chlorophyll A and phycobiloproteins. And it's actually one of those phycobilins, the phycoerythrin pigment that makes them look red. The erythro there means red, so phycoerythrin has a red color. And then if you had that phycoerythrin associated with a protein, that would give you a phycobiloprotein. So what they actually look like kind of depends on how much phycoerythrin they have compared to how much chlorophyll they have. Usually they'll have more of the phycoerythrin and they look red, but actually like the shallower they live in the water, the less phycoerythrin they probably have. So usually the ones that live deeper are more intensely red, and the ones that live less deep might be less red or even not red at all. So in this image here, you can see like here's a rhodophyte, a multicelled rhodophyte that has a good amount of phycoerythrin, which is why it looks red. And then here you have some rocks. On the underside of these rocks, you have red algae living, and if you were to zoom in on them, they look like this. So they're single-celled, and they're actually green because they have more chlorophyll A than they have phycoerythrin, since they're living pretty shallowly. Another thing the red algae all have in common is they have the phycobilisomes, which is a structure inside the cell made out of, well, it's like an organelle, inside the cell that has the phycobiliproteins, so those phycobilin pigments associated with proteins. It's basically functioning the way a chlorosome would function. So it's all these pigments are absorbing photons and funneling them into the photosystems of the chloroplast. So just helping them collect light more efficiently. Red algae are important industrially because some of their cell walls are made of agar and some of their cell walls are made of carrageenans. So it just depends on like the specific species what the cell wall is made out of. Both of those substances are important in food production, and agar is really commonly used in the lab as a substrate to grow, to culture microbes on. So your petri dish would be full of agar, which comes from a red algae. Some of them are called coralline, which means that the cell bodies are actually calcified and they live as part of a coral reef. So they're coralline because they have this mineralized, cell wall actually. And some of them actually live in acidic hot springs, so they're living in really extreme conditions, very high heat and also very acidic. Actually, out of all of the phototrophs that are known, these red algae that live in these acidic hot springs are the most extreme extremophiles out of all the phototrophs. And they actually got the genes that let them do that by horizontal gene transfer from prokaryotes. So the genes that are allowing them to survive at that really low pH and that really high temperature, those are coming from a prokaryote. Then you have the green algae, which actually are closer to plants than the red algae are, which kind of makes sense since, plants look closer to green algae because they have pigments that are closer to green algae. So the green algae are, of course, phototrophs, and their pigments are chlorophyll A and B. So they don't have the phycobiloproteins or phycoerythrin. Because of that, they look green. Plants also will just have the chlorophyll A and B, which means that they also look green, which is a clue that they're more closely related to each other than the red algae are to plants. Most of them are unicellular and microscopic, but some of them are multicellular, and some of them are multicellular and macroscopic, so they're not microbes. It just depends on the alga. And you have them divided into two different groups. So you have the chlorophytes and the cheirophyceans. The chlorophytes are like the biggest group by far, and then you have just a few cheirophyceans. The cheirophyceans are macroscopic, and they're actually super closely related to plants. So, it goes chlorophyte, then cheirophycean, then plant. So on the bottom here, you have different chlorophytes. So you can see some of them are unicelled, some of them are multicelled, some of them form kind of crazy structures. And then at the top here, you have a cheirophycean. So this is actually macroscopic algae, so you don't need the microscope to see it. You could just find this growing. And I probably have actually in the past. Green algae usually live in fresh water. Actually, the cheirophycean that I saw in the past was in a lake. Some of them live in lichens with a fungus. Some of them actually live inside of rocks. Rocks will have little pores inside of them that can have water in there. So you could have like bacteria living inside of there, or these green algae. Those are called endolithic algae, or it's an endolithic lifestyle. Endo inside of, with stone, so inside stone. The rocks are kind of protecting them from the environment. So most of the ones that would live in rock pores, it would be in some kind of harsh environment, or some environment that would otherwise be harsh for them, like somewhere that's real dry or somewhere that's real cold. Actually, in the rocks around here, you can find some green algae. Like if you go hiking in the Gila, sometimes you'll find rocks that have this kind of green coloration to them. And that's actually green algae living inside little pores in the rock, endolithically. It could be just algae in those pores, or it could be a lichen that has the algae and it has a fungus, either way. And some green algae will form colonies where you have like specialization, and some of the cells are doing photosynthesis in the colony, and other cells are just doing reproduction. For the ones that are doing photosynthesis, they would have flagella on them, and they are able to move like together, so they kind of beat their flagella like simultaneously or in synchrony with each other, and that way like the whole colony can move as a single unit. And finally, some green algae produce hydrocarbons, which is, pretty unusual. Actually, this guy at the top here, he has kind of almost like a rainbow coloration. That's actually caused by the hydrocarbons that this species of green algae produces.